It's really nice to see you too, Dr. Adam and Alyssa. Can you hear me?
Yes, thank you.
Yes, thank you.
Great. This is the first time that we have two professors in the broadcasting room for a double live broadcast. So thank you so much for contributing this invitation to us.
Absolutely.
Now, if you don't mind, our sharing will begin in earnest. And today, my guest is Professor Adam Bohnert and Professor Alyssa Johnson from Louisiana State University. Professor Adam is then determined to understand the molecular basis for transgenerational age resetting and to ultimately uncover natural mechanisms by which something old can become young again. And Professor Alyssa is focusing on exploring links between the genetic and molecular process of age-related degenerative diseases. Welcome to our broadcasting room. First, could you please introduce your academic background to our audiences?
Sure, I'll start. So I'm Adam Bohnert. I'm an assistant professor at Louisiana State University in Baton Rouge, Louisiana, United States. I originally got my PhD in Cell and Developmental Biology from Vanderbilt University. And at that point, I was studying just fundamental cell division processes. And then I moved on and did a postdoc in California, first starting at UCSF, University of California, San Francisco, with Dr. Cynthia Kenyon, where we used a really tiny nematode called C. elegans to study the aging process. And then our lab moved just sort of south of the city to a company called Calico Life Sciences that also has an interest in studying aging biology. And from there, then I started my own lab. At this point, it's been about 5 years since I started here at LSU, and we're collaborating actively with Alyssa's lab to study just the fundamental cell biology of aging, genetics of aging, trying to understand ways to help cells maintain health and youthfulness.
Hi, and I'm Alyssa Johnson, and I have a similar track record. So I also did my PhD at Vanderbilt and was studying cell cycle regulation and was focusing on checkpoint pathways. And then I did my postdoc at UCSF in the lab of Gray Davis, and there I transitioned to working in fruit flies, which is what my lab works on now. And there I was interested in studying neurodegeneration, genetic causes for that, and creating and developing disease models that we can use to study the pathology and also maybe test some therapeutic strategies. And there I became really interested on the process of autophagy and lysosomes. And then I also started here at LSU about 5 years ago, and my lab continues to use fruit flies as the main model system, but we've also, in collaboration with Adam's lab, we've also been doing some work in C. elegans as well.
Thank you. Now our audiences may have a little knowledge about your direction. Well, before the start, let me do a little gossip. Since you're in different lab, then after work, will you discuss the work-related matters or help each other with the research?
Yeah, so yeah, absolutely. So our labs are basically, we do a lot of joint, lab meetings and things like that. So we're always sort of discussing ideas in our labs. The members of our labs are collaborating and sort of always sort of interacting with each other. But yeah, we just given our similar, research interests, there's a lot of just sort of natural collaborations that have sort of come out of, but the tubular lysosome projects have been sort of the main collaborative focus. And then we each have some other projects that are sort of, independent of each other. But this is something that, we've been collaborating on really from the beginning.
And I have to say, you're really in an ideal state of scientific life. Now, let's get out to the business. My first question is that a dietary restriction has been recognized as one of the most effective methods to enhance lifespan in animal models, such as C. elegans. In your recent study, your team also embraced the concept of dietary restriction. And first, could you please provide a brief overview of this concept?
Yeah, so dietary restriction is essentially just the practice of limiting food intake without actually malnourishing the body. So limiting it to a level that isn't going to deplete the body of sort of essential nutrients. And as you mentioned, there's a lot of evidence that this can have positive effects on organismal health and even extend lifespan. And this has been shown in a number of model systems ranging from worms to mice, and now there's even clinical trials in humans and that are showing beneficial health benefits. And so the basic idea is that you limit the amount of calories that you're eating, in providing your body with periods of fasting, and this can be done in different ways. So some practices you just reduce the total number of calories that you're eating in a day. There's now sort of other practices where you limit the time of day that you're eating those calories, and this is called time restricted feeding or intermittent fasting. And that also has been shown to have just as much benefits as reducing the total number of calories. And so you can essentially eat the same amount that you would normally, but if you just restrict that to a smaller portion of your day so that you still have a period of fasting at some point during the day, that can still provide beneficial effects.
And how does the dietary restriction contribute to extending lifespan?
So that's something that we're really fundamentally interested in, and we know that the major cellular process that gets triggered is a process called autophagy. So when you're fasting, and your body is depleted of nutrients and it's not getting those nutrients from sort of a food source anymore, your body tries to find some sort of other way, alternative mechanism to create a pool of nutrients. And so autophagy is one mechanism that it can do that. So autophagy is this cellular homeostasis cellular process, and it has two major functions. And the first is to scavenge for molecular damage. So protein aggregates, damaged organelles, things like that. It will scavenge your cell to try to clear those from the cell so that it doesn't cause damage, cytotoxicity, right? But it also has a second major function in that when it clears that molecular cargo, it's also delivering it to the lysosome, which then recycles, breaks down that cargo into smaller pieces like individual amino acids and lipids, and then recycles that back into the cytoplasm. And so that provides now your cells with a source of nutrients that it's not getting from the food source. So when you're undergoing dietary restriction, it triggers autophagy mainly to try to provide an alternative source of nutrients. But in the process of that, it also needs some kind of substrate. And so it's going to go around your cell to try to collect any sort of molecular damage in the cell. And that's really what provides the health benefits, because you're sort of cleaning up the cells, by triggering autophagy through this mechanism.
Thank you, Dr. Alyssa. Actually, our audiences are the ordinary people that have so much academic background on this. So first, may I invite Dr. Adam to explain for us what exactly is a lysosome and what is its primary function?
Yeah, that's a great question. So as Alyssa was mentioning, we're both really fascinated and interested in the lysosome as a part of a cell, right? So the cell is the most basic unit of life that we think of, and a lysosome is a compartment that's inside of our cells. And Alyssa was just mentioning, this process of autophagy, whereby the cell takes material and recycles or degrades or turns it over. And the lysosome is really famous for executing this function. So if you look at the name lysosome, it's lysing, it's degrading material in the cell. And so it's enclosed within its own membrane. So it's separated out from other parts of the cell. It's a discrete compartment. It's actually very acidic within this compartment, which is different from of the other parts of the cell. It's at an acidic pH. And this is important because all of the enzymes, the molecular players that are within a lysosome that are degrading things, right, that are cutting things up, that are chopping things up, that are digesting things, they require this acidic environment to be active. And so what happens is that this material that's going to be cycled, it gets by a variety of mechanisms, it gets shuttled to the lysosome. And then the enzymes that are within the lysosome can then cut this up and recycle the material. And in the process, clear damage. Right. And so it's a centralized location to potentially take in waste and to then turn it over.
That is also the way that links lysosome toward the aging process.
Yeah, and so the thing that's interesting there is, one big question in aging research is really at a cellular or molecular level, what is the initiating domino? What's that first domino that's falling that's causing everything else to deteriorate? And so, there are obviously many parts of the cell, but one thing that's been seen in a variety of systems, including single-celled yeast, is that a very an initiating event in this deterioration seems to be dysfunction of the lysosome or dysfunction of the autophagy lysosome system. And, this is obviously problematic as we age if our lysosomes are not working, because then the molecular waste that is there that maybe needs to be cleared, it's going to start building up and it's going to start accumulating. Okay. And so if there was a fundamental way to come in and perhaps therapeutically or some intervention to boost lysosome activity, that would be very beneficial, because then we could, with aging, take a cell that's maybe accumulated some damage and help to take out the trash, to help clear some of this damage such that it's maybe not as toxic as it would be. And so that's, within our labs, that's a major goal, is to find ways that the lysosome can maybe naturally become more active, can become more proficient, because we think this could be an entry point to improve healthy aging.
So when we enable lysosome, it may help us to clear up some of the waste in our body so that we could counteract aging better. Is that way?
Exactly. And that's our recent study that we published together. One mechanism that we found that we think is important here is that this compartment, this lysosome compartment, is actually changing shape. That, typically it looks like just like a circle, a sphere.
Spherical.
Yeah, just a big circle, and these circles that are dotting the cell in different places. But what we found is that in the context of dietary restriction, now this shape, it really changes, and you get these branches and these tubules that are coming off of these vesicles, and they're forming a very interconnected complex network. And we have some evidence that by making this really expanded tubular network, that's aiding in this digestive process of autophagy, that it can help the cell to support more turnover of material during the dietary restriction.
Then I may propose a question first. What is the distinction between the spherical and tubular lysosomes in the context of aging research? Dr. Alyssa.
Yeah, so until recently, there had been some reports of tubular lysosomes in other contexts outside of aging, like in the immune responses and things like that. And it had sort of thought that, they served specialized roles in immune cell function and that they weren't actually autophagic. But we and others have now demonstrated that in certain cell types, these tubular lysosomes are in fact autophagic, and they can degrade cargo. So we've demonstrated in our recent paper that if you look at the rate of turnover, so the how fast they can degrade and sort of take up this molecular damage, sort of the cellular trash, the vesicles do have capabilities of doing that. So they can degrade cargo. That's not that they're not able to, but when you have this expansive network, you get much more rapid turnover and degradation of this cargo. And so the model that we sort of have is that when you have a huge autophagy need, so when your body is under starvation conditions, for example, or dietary restriction, and it needs to ramp up autophagy, you're going to have all of this, this larger autophagic demand now. So there's going to be a lot more things that it's going to need to try to degrade. And the vesicles just aren't really efficient enough to do that. And so as a response, it will create this tubular network that spans the entire cell, and it provides really a bigger surface area for the lysosomal compartment. So you can kind of think of it as a biological grid that now you don't have to have, these individual autophagisms trying to find these little vesicles around the cell, but there's a huge platform now. And you're just going to really increase the chance that an autophagosome will encounter a lysosome and be able to fuse and degrade that cargo. And so it just provides basically a platform for greater autophagic degradation.
Yeah, and if I can piggyback off of Alyssa here a little bit, is the model is, if you think about like your own house or some building that you live in or work in, and maybe there's an individual trash can in every fifth room or so. There's not a trash can in every room. There's maybe a unit where you can dispose of some waste here and there. And obviously, maybe there was an overload to the system, and for whatever reason, you had a lot of trash to get rid of. Well, you would have to maneuver to that location. Maybe the trash would be full. Maybe there just wouldn't be the capacity to handle it. But if instead you had a trash chute, almost, right? Almost some tubing of a trash chute that just extended from room to room, and you could access easily sort of on demand. That's going to, reduce the bottlenecks. It's going to increase access. And we think there are probably other things that these tubules are doing too that are helping the degradation that we don't have as strong of evidence for yet, but we're interested in looking at it. But it's one way to think about it, of how having this sort of bigger, broader network to receive the cargo, we think could enhance the turnover efficiency that's happening at this lysosome.
So the trash, when it is overloaded, we cannot handle it by ourselves. We need to turn to others for help.
Yeah, and I think that, having, the communication between the lysosomes as well, creating this continuous network, also aids in the efficiency that they can also degrade things too, because, the vesicles, they will reach a capacity, as, Adam was saying, that, if once they, take on so much cargo, they have to go through a process of sort of rejuvenating to become a functional lysosome again. But if you have this continuous network, they can kind of share the burden then, across a, larger pool.
What are really amazing is that your study suggests the longevity effect triggered by lysosomes can be inherited by the next generation. What is that mechanism behind this phenomenon?
Yeah, I can take that. So, there is work done by other groups besides us that have historically looked at this inheritance of longevity, right? And obviously there's an inheritance of longevity at the level of genes, but there can be an inheritance of longevity in response to environment as well. And a very famous example is starvation, okay? And that if you look of descendants of animals that have been starved, even if those descendants are themselves well-fed, right, but say their grandparents were starved, that original starvation event a few generations back is going to have an impact on the health and physiology of the descendants that are now fed, even though they themselves are not starved. And so, there have been many groups that have studied this, and what's been shown in the C. elegans model and the worm model previously is that if you look at these fed descendants, that they actually live long. okay, that there's some change to the signaling or some change to their physiology in response to their ancestors being starved, that now they're living long as a result of this. And so, yeah, we have some evidence from our work now looking at these tubular lysosomes, which we see as responding to a starvation event, responding to a dietary restriction event, that if we look not only at the starved animals, but at their descendants, right, the animals that are maybe 2 or 3 generations out, that now we see these tubular lysosomes already induced in these fed animals, right? We normally don't see them induced in the fed animals as much, that where they're constitutively well fed. But it seems that if there was a starvation a few generations back, that the signal that's stimulating the tubular lysosomes to form, that it's still there a few generations out. And what we found is that if we prevent that from happening, if we prevent the tubular lysosomes from forming in those fed descendants of the starved animals, that this effect on longevity no longer happens. And so we think that there's some part of this, it's some part of the mechanism, right? This change to the lysosome morphology that is important for this transgenerational inheritance of this longevity effect. We're still not really fully clear on the mechanism by which it does it. We think it's partly due to how the tubular lysosomes are acting or what they're doing to support autophagy. But we're still trying to understand that in a little bit more detail.
And in the last, or as it was the last before last broadcasting, we actually have reported some of the desserts that focus on the reproductive system. It has seems that some of the characteristics that can be inherited from parents to especially some of the longevity characteristics. But at that time, we only focus on the reproductive system. We don't have imagined that lysosome also follows the same rule or have the same effects.
Mm-hmm. Yeah, and so it's really it's an exciting thing for us too to think about and to consider, because what our research shows is that there are genetic influences on longevity, but there are also non-genetic influences, right, that can be passed on, that can be transmitted in response to environmental changes. And so we think in this case, there are some traits that are, obviously being passed on that are enhancing the health or vitality of these organisms that's stemming from a starvation event, but the starvation event actually happened 2 or 3 generations ago.
And Dr. Alyssa, I'm also interested in another aspect that regarding the transgenerational impact on longevity, is there a time limit? In other words, if the offspring doesn't undergo dietary restriction, and when does this longevity effect disappear? Will it disappear?
Yeah, so what we found in our studies is that there is a limit in terms of the number of generations that the effect gets carried out to. And this has been shown also for the beneficial effects of dietary restriction as well, that once you get out to about the 5th generation past that original starvation event, the effect isn't seen anymore. And what we've also seen is that when we look and actually sort of quantify, the amount of sort of networks that we see, tubular networks that we see for the lysosome, that the effect does diminish with each generation. So the first 2 generations after the starvation event, the animals still have really robust tubular lysosomal networks, but then by the 3rd generation, it starts to diminish some. So there is somewhat of a dilution effect with each generation. But within each generation, animals that have really robust networks, it doesn't seem like there is a major time limit on that, and that those beneficial effects will actually persist for that individual's entire lifespan. And so we did this experiment actually where we looked in the 2nd generation, so 2 generations after the starvation event. And there we saw some heterogeneity, so some differences in the sort of how well they were able to, form these tubular networks. So some animals in that generation had, really, really robust networks just as much as the originally starved parents or grandparents, right? There were others in that same generation that maybe had weaker ones, that they had some formed, but they weren't really as robust as maybe the original starved parents. And so we actually did an experiment where we compared the lifespan of the animals that retained really strong networks versus those that did not. And we found that there is, the animals that retained really strong networks lived on average longer than those that did not. So there is a correlation there with how strong and robust these networks are and the longevity. And so, but I don't think there's a time limit. If you look at sort of the individual animal, it seems like those beneficial effects do persist for their entire lifespan, but there is a dilution effect that occurs with each generation.
When we change another animal model, will the things happen again, do you think?
Yeah, so there have been, evidence for transgenerational, inheritance of, similar sorts of things in mouse models. It's a little bit harder to really study this, obviously in humans, but there's been some correlative studies that have been done. But, so we do think that this at least translates up to mammalian, model systems. And, they've shown certain, similar kinds of transgenerational benefits that can be inherited in mouse models.
And besides dietary restriction, are there any other methods to activate the tubular lysosomes and promote lifespan extension, such as medication or some other methods?
Yeah, so we know we've done some genetic studies, and we know that at least inhibition of a gene called mTOR is sufficient to induce these tubular lysosomes even in well-fed animals. So it sort of mimics the dietary restriction effect. And so mTOR is a major nutrient sensing protein in our cells. And so there are drugs that are targeting mTOR like rapamycin. And so we haven't directly tested that in our system, but we suspect that, things like rapamycin would also induce tubular lysosomes. And we also, found another genetic way to induce tubular lysosomes constitutively. And we can do this by overexpressing another small gene called SVIP. And when we do this, we're able to create these networks, all the time, even if animals are really well fed. And so one of our, long-term goals is to try to target SVIP as a therapy. And one of the kind of cool things about SVIP is that it's a very small protein. It's only 77 amino acids. So it's a very, very short peptide, which is something that could, in theory, be used to synthesize a small peptide that could be delivered sort of like a small molecule drug.
And Dr. Adam, could you please elaborate on the SVIP protein?
Yeah, so we're really excited, I think, about SVIP. And so this was research that Alyssa had started looking at SVIP during her postdoc, and it was studying it because there was evidence from the fly system that this was a key regulator of the tubular lysosome network in this animal, that if you got rid of it, that the tubules didn't form as well, and if you added it back, the tubules just formed very well, very strongly. And this correlated with physiology, that the animals that were lacking SVIP seemed to be a little sicker, showed some degenerative phenotypes, whereas those that had SVIP added back seemed to be very robust. And so one thing that was curious when we started doing this with the worm is we obviously wanted to test in the C. elegans nematode if SVIP could also have a conserved role in regulating these lysosomes. The thing is, though, if you look at the genome of all the genes that are listed for C. elegans, there is not an annotation for an SVIP gene, okay, what we would call a homolog of the fly SVIP. There's not anything that's been identified in the worm to be an SVIP gene. So we could not manipulate that gene in the worm system. Instead, what we did was we added back to the worm system the fly gene. Okay, so what we were doing was we were taking the gene that encodes for SVIP from the flies, and we were having the worms now make this protein. And when it made this fly protein, that's when we saw that these tubular lysosomes were constitutively induced. What we now know is we can take the human version of SVIP and also make the worms express that. And we have some encouraging evidence that I think this is also inducing tubular lysosomes in the animal. And so, yeah, we're really excited to try to see if there are any really key parts of SVIP that are stimulating this, because even though it's a very small protein, if we can get it even smaller, it may be more powerful as a drug or amenable as a drug to help.
It targets it.
Yeah, to do that. So we're trying to understand what parts of SVIP have these capabilities, and if we can, potentially develop it further. If not, there are certainly really interesting aspects of biology that we can learn from it.
Then actually you have mentioned about the gene playing a role in this process. Then I want to know how much genetic factors, do you think that plays a role in extending lifespan?
Yeah. So that's a great question. And, we talked some already about genetic versus non-genetic factors that could be contributing, right? So obviously there's influence of genes. There's also influence of lifestyle and environment. Obviously there's going to be this complex effect of all of these coming together. But there is a really, now a well-established basis of longevity in genetics. So, one of the most famous examples comes from my postdoctoral advisor, Dr. Cynthia Kenyon, where she had shown, I guess it was about 30 years ago now, she showed that you could have a single mutation in one gene that was called DAF2. And what this is it's an insulin receptor gene. Okay, you could have one mutation in this gene, and the tiny worms lived twice as long, and they were very healthy as they were aging. And one of the questions, I think, when this was first discovered was, you're looking at this lifespan or aging regulation and a tiny nematode that normally lives 3 weeks, and instead you're making it live 6 weeks, right? And
But when it comes to human,
yeah, the question was, how does that relate then to humans? Is that meaningful to humans? And what's been shown now with this, huge improvement to genome sequencing and genomics, right? We can really understand genetic variants in individual humans, okay? And so there are individuals that live to be over 100 years old that are, and we call these centenarians. And so there have been studies to try to look at centenarians to see what genes are maybe variants that these centenarians would have that could maybe contribute to their long lifespan. And what's been found now that's interesting is that the homologs, right, the same gene that I just discussed in the worm that was mutated that helped that worm to live twice as long, it's now known that same gene shows variation in these long-lived humans and these naturally long-lived centenarians, right? And so there's a very, very strong evidence that there is a conservation of gene function in the control of longevity, in the control of aging, and that even by studying, what you might think of as simple animals like the nematode C. elegans or the fly, that we can learn really, really fundamental things about what genes are controlling aging, right? What genes are controlling longevity. And so from our perspective, we're really interested in identifying genes that are controlling the lysosome. And I certainly think that there are genes that are impacting lysosomal function that are key longevity regulators. And so we're getting at it with that focus, although certainly there are other genes that are impacting other cellular processes that are, equally, if not more so, important too.
And Dr. Alyssa, Dr. Adam just actually make a comparison with C. elegans and human about their longevity impacts. Then if we aim apply the principles of dietary restriction from worms to humans? I mean, on the aspect of longevity, what factors should we consider, or what are the differences between the two?
Yeah, so I think there are, a lot of, the effects of dietary restriction have been shown to be really well conserved from, C. elegans to humans. And so, the benefit of being able to study the effects of dietary restriction in these simpler experimental model systems is that we can really try to get at, what are the genetic factors that are being triggered by this sort of environmental, behavior, right? So how does dietary restriction actually, create healthier individuals? And we know that it, even though you're not making genetic mutations in the animal, it is still influencing how the genes in your body are getting expressed. And so just by altering your diet, you're getting changes in certain genes that are being expressed or turned on or off. And we can understand, that from, the perspective of trying to study this in these experimental model systems. And all of these genes that have been found, they all are conserved in humans as well. And so I think it is likely that, the same pathways are being also triggered in humans. And the fact that we see the same sorts of physiological health benefits translating into humans would suggest that they're probably using very similar genetic mechanisms to sort of achieve that.
Then you have just mentioned about the mTOR also played a role in the healthy aging induced by the tubular lysosomes. So my question is, what are your perspective on other substances related to mTOR, such as rapamycin, and what do you think? So it has a promising future for us humans to improve our lifespan.
Yeah, so I mean, I think rapamycin is a good example here, and there are many other groups that have a strong focus on rapamycin and know rapamycin a lot better than we do. But one thing that I'll mention is that's really, I think, interesting is that there is this push within the aging field, the aging research field, to try to understand that if we start thinking of treating aging, what does that really mean biologically, or how would we go about assessing that, right? Is it a combination of diseases that we're looking at? What are we looking at as a treatment of aging? What does that mean? And so things like rapamycin are starting to push us in this direction to make us think about how would we assess something as maybe a anti-aging therapeutic or anti-aging molecule as we start moving to humans. And so, rapamycin is now being tested in some larger animals. So there is a project that's called the Dog Aging Project, where scientists.
Dr. Matt Kimberlin.
Exactly. And so scientists are enlisting really dog owners around the world to become citizen scientists. And it's a very large study, but part of this is involving some rapamycin treatment or rapamycin therapeutics to try to see effects of this using a large animal. So I think that there is really some exciting data out there and some exciting thoughts on how rapamycin may be applied. There are other, if you want to think about it from a drug standpoint or a compound standpoint of something that's anti-aging, the other one that gets a lot of attention is metformin. And there are human trials now with metformin. It's called the TAME trials of targeting aging with metformin. And these are also really interesting and trying to see what the results are here and trying to see what some of these compounds are. And so, from our perspective, with our research, we're obviously interested in how these different molecules that are known to affect longevity in a variety of species, including perhaps humans, how these could maybe influence our tubular lysosomes. We don't fully know that yet, but that's something that we're interested in looking at. And we're obviously very interested in trying to see if there may be other small molecules or something novel that's perhaps more specific to the tubular lysosomes that we could utilize, perhaps something based on SVIP, to see if we can manipulate this in a therapeutic manner.
I also remembered that Professor Toren Finkel, he has published a paper last year, I suppose. It also focused on lysosome. How does it functions to help the model animals to live longer? And he also found a new pathway called PITT, PITT, a name after the Pittsburgh University.
Mhm.
And how do you exactly on that?
Yeah, I mean, I think so at this point, how that feeds into, say, the lysosome tubulation, we don't know. One thing that I think Alyssa and I would both say is that, historically, the lysosome tubulation was kind of thought in the sort of autophagy field, the degradation aspect of lysosomes, to almost be like an artifact or a very rare event. And so even if we go back and we look at historical papers that are published on lysosomes, if you look at pictures of the lysosomes in some of these, say when some of these other pathways are manipulated, you can actually see evidence of lysosome tubulation, but it's never really discussed. Because again, I think it hasn't been recognized that this is a fundamental way to manipulate lysosome biology to impact autophagy. And we're still seeing this as we read lysosome papers. So with that pathway in particular, we're not really sure how that would feed into some of the factors that we're talking about. But we're really interested at trying to look at the crosstalk to try to see if there are sort of interactions among, really certainly unique lysosome pathways that could be feeding into and controlling lysosome tubulation. And I think it's likely that probably many pathways converge on this, since it seems to be a fundamental response to some stimuli such as dietary restriction.
Last month, we have met Dr. Toren Finkel on the ARDD at Denmark, at Copenhagen. In the interview, he also told us that he is not so sure. It is still undergoing the research whether the pathway PITT can help in mice, the model animals to evade from the degenerative diseases. It's still under discussion and research.
Yeah, and I think, with ours too, Alyssa, you may want to comment on this a little bit, but, Alyssa has, I would say, from her fly side of things, maybe more evidence with the lysosome tubulation that, we think that the lysosome tubulation, does have some role in sort of modifying sort of neurodegenerative or degenerative, pathologies. Alyssa, if you.
Yeah, so one of the things that we've been interested in is seeing, what sorts of cells have them. So, one thing I want to, point out is that not all cells make tubular lysosomes as far as we can tell. And then, in certain cases, some cell types will only do it upon, some kind of stimulus like a starvation event. But, for example, neurons, we don't really see these tubular lysosomes forming. But one of the things that we have shown in this paper and also another paper is that if you induce tubular lysosomes in one tissue, it seems to benefit the whole body. And so it can even benefit tissues where you're not getting these tubular lysosomes induced. So for example, in C. elegans, we found that if you induce these tubular lysosomes in the gut, so in your gut cells, it can actually provide improvements in muscle tissue health, and it can slow down the rate of sort of muscle deterioration. And so there is this cross-section. So even if we're not manipulating muscle tissues at all, just inducing them in the gut cells has beneficial effects for so slowing the rate maybe of aging of other tissues systemically. And then another example in flies that we've shown is that if you induce them in muscle cells, it can provide support for motor neurons. And so if we have a disease model where we've induced a mutation that causes a motor neuron disease, and so you'll get degeneration of these motor neurons, so the neurons that are going to be involved in sort of how you move your body and things like that. If you induce these tubular lysosomes to sort of a higher level in muscle cells, it can actually rescue the motor neuron degeneration, even though we're not doing any kind of manipulation to the neurons themselves. So we think there is, a lot of exciting potential for trying to trigger these things in certain tissues of the body, and it could maybe provide, beneficial effects for all the tissues in the body.
That is exactly what our audiences are focusing on. They in our broadcasting room, they also propose some questions. The first question is, besides the health benefits, what can the lysosomes normal function can provide us for benefits? What you have mentioned is benefit for our muscle and our brain and some other things.
Yeah, and I think that, all of these, the tissues in our bodies, we sometimes think of them, as a cell as being an individual, unit that's separated, by these structures that prevent it from, communicating with other cells. But that's actually, I think not the case. There are mechanisms that allow the individual cells in your body to actually communicate and trigger certain processes, and so there's a term that's been used called teleproteostasis. And so proteostasis is this process of degrading proteins in your cells, and it's been shown in different systems that if you trigger proteostasis in one tissue, that it can also stimulate proteostasis in distant tissues, other tissues where you're not making any kind of manipulation. So I think there is a lot of crosstalk between tissues in our body, and there's, we still don't really understand a lot of the signaling mechanisms or cues of how that happens. And there seems to be certain tissues that are better at it than others. So the gut cells, seem to be a really sort of centralized tissue in our body that seems to communicate nutritional needs to the rest of the body. And so I think that's why things like dietary restriction have such a big systemic effect, because you're affecting this tissue, so that's sort of a central part of your body that then can communicate the nutritional needs to the rest of the body and kind of stimulate maybe autophagy in these other tissues to try to, create, bigger pools of nutrients.
Yeah, and one other thing that I'll say related to your question is there is evidence from several groups, including Anne Brunet's group at Stanford, that keeping sort of a good lysosome activity is very important for stem cells. And we know of stem cells as being critical for regenerative potential and sort of repair. And so this is one specific example of a really important, I would say, cell type where maintaining really healthy lysosome function, you could obviously think of that impacting then regeneration, repair, and health at a different level.
So, just you have mentioned about the stem cell, then I've certainly got another question is that if we want to extend our lifespan, can we count on the stem cells or to do some medication on that, or that there are so many stem cell therapy,
Yeah.
their discussion or have already been matured to be done by normal people, and what is your opinion on that?
Yeah, I mean, the one thing I would, I guess, point to in regards to that question is there's a lot of cutting edge research in terms of in vivo reprogramming. So sort of reprogramming your cells to be more stem cell like. And, there are groups that can do this in animals. And when they do this, there are effects on lifespan and on aging and obviously on regenerative potential. And so I think, the simple answer is, I think the stem cells are part of this, that if our stem cells are exhausted or no longer to function in an aged animal, the capacity for really a healthy lifespan is, I find that to be minimal. I find that to be, unlikely. But yeah, there are a lot of things that go into it, and I think there are some therapies that are in the works that could be applicable to improving stem cell function with aging. Another sort of, I would say, unique intervention that's been looked at is parabiosis. And parabiosis is when you take, say, two animals and you join the circulatory systems of these animals so that they share systemic factors in the blood. There is a lot of historical research of one animal getting effects from the other in this pairing, right? And if you take a young animal and you take an old animal and you join them in a parabiosis pairing, the old animal will start to show enhanced stem cell activity, and this will impact tissue regeneration. It's also thought to boost cognition, right, in the brain by influencing sort of brain activity, brain health. So yeah, there's probably some molecules that are sort of youthful molecules that are within these systemic factors that people are trying to identify. But yeah, I think that one likely avenue that could influence longevity research is trying to identify these things that affect stem cells. Sure.
Thank you so much for the detailed elaboration on the your research and other theoretical problems. Now, maybe I will propose some other questions that is more practical, that's proposed by the audiences in our live broadcasting room, since that they focus more on the questions that related to their daily lives. I translate to translate their questions to you.
Okay, great.
Then, Dr. Alyssa, the first question is that the differences between people with poor digestive function and normal people and at the aspect of caloric or dietary restriction?
Yeah, so I don't know if this has really been studied and if they have poor intestinal function. We do know that dietary restriction itself can actually improve intestinal function, and so dietary restriction can activate intestinal stem cells that will help maintain and create a better sort of digestive system. So it could be used actually as a way to maybe improve, digestive deficiencies, or if there are certain things that people have, dietary restriction could likely be a mechanism to try to improve, dietary function. Whether or not they would have reduced health benefits because of that, I'm not sure if that's really been tested experimentally.
And how does the calorie restriction ensure nutrition intake?
Yeah, so
could we?
So there is a fine line, right, between reducing calories so much that you're then in a state of malnutrition. And so it is important to consider that if you are doing dietary restriction practices, that you want to make sure that you're still getting the essential vitamins and nutrients and those sorts of things, taking, additional supplements maybe to make sure that you're getting all of the, essential nutrients that your body will need. And so, I think one of the more interesting dietary restriction practices is this time restricted feeding or intermittent fasting, because in these practices, you can actually eat, fairly normally, but just restrict it to sort of a smaller portion of your day. And there was a study that was done in fruit flies recently that was published in Nature that if they restrict it to the beginning part of the day, so the first 6 hours of the day, the flies can eat just as much, same exact diet as flies that are, eating, sort of normally, and they would show the same lifespan extension and health benefits. And you can actually do that not even every day. You can just do it every other day and you would still see the same sort of benefits. So it isn't that you really have to commit to doing this every single day, but even just having, certain periods of fasting it provides, beneficial effects. And I think there's evidence for this, in
intermittent fasting, right?
Yeah, intermittent fasting. Yeah, and I think there's evidence for this even in sort of cultural practices. The religious practices that, do fasting as part of their sort of, religious practices, they also, see sort of, similar health benefits. And that's been kind of, known sort of historically.
And what do you think, or in your experiences, how to do the fasting is the best way for us human beings?
Yeah, I mean, I think there are a few things, and so it's interesting to think about this because as a scientist, as you're studying these things and you come across certain findings and you see how it affects animals, it sometimes makes you question some of your own lifestyle choices and what you're eating for dinner, and how you're eating and how you behave, right? So, personally, I don't do intermittent fasting. I know a lot of people who do, and I have friends and family who do, who will say it has a great effect on how they feel. I think there are other lifestyle things that also have positive effects that have been proven, which is exercise being a really key one. You'll see really a large effect of exercise on sort of health, obviously, and this can impact longevity. So I'm not sure that there's necessarily like one single biggest change that could do it. But yeah, there are, I think, additively some small lifestyle choices that can be made that can certainly make you, feel healthier and be healthier.
And I'll just to add on that, I think, there are, evidence that, combinations of things like dietary restriction and, maybe improved exercise and, can have sort of a compounding effect. And another aspect that can have a compounding effect with dietary restriction is sort of your circadian rhythm and sleep. So, what they, in one study, they showed that, it's better to sort of keep your calories kind of at the beginning of the day and have the fasting period later in the day before you sleep, because we know that autophagy is also naturally more active while we're sleeping. And so if you have that fasting period before you fall asleep, you sort of kickstart autophagy. And then when you're sleeping, it sort of boosts it to an even higher level than it normally would have. And so you can actually, create an even, greater effect in autophagy activation in that way.
But fasting, how many days or how long time can induce autophagy in our human being?
So I think, some of there's, now there's all these different kinds of, diets, like, where you fast for the first 6, or you only eat in the first 6 hours of your day. And so then you have, another 6 hours of fasting before you fall asleep. And then you're obviously fasting while you're sleeping as well. There's, others where you eat one meal a day within an hour, and that's probably the most extreme, version. I think, I think having any sort of, extended period of fasting, in your day is going to be beneficial. And maybe there's just different, variations in the total effect size that you might have. But, again, you want to balance, sort of the fasting with making sure that you're not going to be, malnourished in the process too. And I think doing these fasting practices intermittently, every other day or something like that actually, has similar health benefits. And it's probably going to help, one, make it more practical, I think, for people to actually do and be consistent with, because, I think, most people enjoy eating. And so if you do this at least every other day, I think it's going to be something easier to actually stick to long term, rather than doing it, really extremely, and then, going back to sort of like older, like, habits.
If we need to continue to do it every day, I suppose not so many people can stick to that.
One thing that I wanted to add to your point of sort of, I guess, scientifically, is with the animal models that we use. So like with the worm, we have fluorescent markers where they appear different colors depending on if autophagy, the degradation of the lysosome is happening or not. And so we can take the worms, and usually the worms are constantly on a food source. So we have them on a bacterial lawn. They eat E. coli bacteria as their food source. And we can take those worms and we can move them off of the food and move them to, say, a plate that doesn't have this bacterial on. And if we're looking at those fluorescent markers, we can see those fluorescent markers start changing in terms of the color within, a couple of hours. We can start seeing this happening. So it is a pretty fast switch that will happen in terms of the metabolism as these animals are removed from food. And so, yes, if you do have if you have this response, right, or if you're sort of having some sort of intermittent fasting, you can achieve some sort of activation of autophagy with a variety of, I think, regimens. It's just depends on how it's organized.
And do you have a plan to continue this program or this research? What is the next step of your research?
Yeah, I think, it sort of immediately we're really interested in trying to identify more molecules that, we could target to induce, tubular lysosome sort of on demand, right? In a way, because, I think ultimately the goal, a big goal in sort of people that study dietary restriction from the biological sense is to try to find starvation mimetics, things that can mimic the same, beneficial effects of dietary restriction without actually maybe having to practice dietary restriction in our life. And, I think that's a goal for a lot of people. And I think there's obviously a lot of public interest in that as well. So, I think the way to do that is to figure out what are the major factors that are controlling the induction of tubular lysosomes or autophagy, or, we know a lot of the, downstream pathways now that are triggered by dietary restriction. Can we now go in and target those synthetically and try to trigger, these pathways without having to manipulate the diet of the organism?
Yeah, and I would just say, building off of that, so that's, I think, understanding some of the fundamental biology and then starting to apply it to perhaps increase health. One thing that we're also really interested in this regard is really testing. Alyssa was mentioning some disease models, trying to understand if we can apply this strategy not only to promote healthy aging, but to perhaps combat some disease models, or to lessen sort of the pathology that we would see, or to improve the health of the animals that have some sort of perhaps disease mutation. If we induce this type of a response at the level of the physiology, can it also combat certain diseases? That's something that we're interested in looking at, that I think goes hand in hand with looking at how it affects the normal aging or sort of healthy lifespan of an animal.
Yeah, and for degenerative diseases, a lot of neurodegenerative diseases, the major sort of biomarker for neurodegenerative diseases is an increase in protein aggregates or molecular damage. And we know that a lot of degenerative diseases have mutations in the autophagy pathway that can trigger these disease phenotypes. So if we can find ways to maybe bypass those defects, potentially by activating tubular lysosomes, or trying to create, alternate routes of activating autophagy in those disease models, that could provide, some therapeutic strategies.
I also think that it's a very promising future for us to make the results of this research, to convey it from the elegans to other model animals to human, and also convey the same effects from longevity to aging-related disease. It has a very promising future.
Yeah, absolutely.
And although not willing to, our interview's need comes to an end. It is really appreciated that you accept our invitation and share with us very detailed and fruitful interpretation on this experiment. And also, I hope that we could get close contact in the future, and I'm very looking forward to see more scientific research results that produced by your cooperation.
Great, thank you so much.
Thank you so much.
And let's call it a day.
Great. Thank you.